Good afternoon, Lindsay.
Good evening.
Jeremy. Test your speaker. Jeremy, do you want to test your speaker?
Yes, can you hear me?
Oh yeah, can. Lovely, thank you. Bill, can you hear me?
Yeah, I can hear you.
Good, thank you. Hello, Connie.
Good morning, Lindsay. Jeremy.
Morning, Connie.
Morning, Bill. I'm just looking for all our speakers.
Yeah, I was just looking. I think Alexander's on. And are you with us?
Hello, I am. Hello.
Hey, there you go.
I know it's very early for Chantineau.
Haven't seen Shantini yet.
Nick is fashionably late, I think.
Ways.
Well, it must be what, 6 a.m. for him?
Oh no, what did happen? Oh my god.
von der Voice zur Arbeit 12 Minuten.
And out so far.
Claro. Por acaso nós fomos lá passar um fim de semana meio foi bem 3 ou 4 vezes a Isabel.
I see Australia's on. It's very nice, even it's silly o'clock there.
Não, o que eu comprar, ou seja, tem que fazer até a casa do meu. Comprar o que veio tudo e nem literalmente a única alteração da casa tem grande. A mesa era para assim, aquelas mesas de madeira mais ideal, para mandar fazer aqueles grandes brancos. Em cima, usou em cima da mesa. É literalmente a única alteração. A única peça que a casa não tem de origem. Ou seja, a casa agora tem que se fazer.
Could I ask that you put your speakers on mute, please? Thank you.
A lot of people.
A lot of people.
I think we'll wait for Shantanu since he's doing the welcome, at least for
Yeah.
a few more minutes.
I think that's a good idea.
Isabel já
Willem, we're picking up.
Vai for uma festa. Também fora.
Possibly. Willem, we can't hear exactly what you're saying, but
You too, Tim.
I assume it's the airport that he's at or something.
Yes, he is. Yeah.
well, it looks like we have great participation, 99. We can go over one more. Oh, there
There
comes
we
the
go.
100. Excellent. So this is our 4th educational session, and we've had great response, and we're glad you're all here.
Yeah.
So could you ping Shantanu?
Okay, Lindsay, I just pinged him.
Oh, well done. Thank you, Samantha.
So while we're waiting for Shantanu's welcome, those of you that have seen the program, we have 3 dynamic speakers today that I hope will really fill you in on what's going on in the background with this study and give you some insight. And I really encourage everyone to use the chat to ask questions, and also to raise your hand to ask questions. And that's why we're here today, to hear your questions, and also for the team that's working on the analysis to give you some insight on what's happening. I think one of the surprises to me anyway was the amount of work that goes into the finesse around these probes. And Jeremy certainly will fill us in on some of that, and Bill also. So part of this whole validation process is actually choosing the probes and getting them to work. And Shantanu,
Yes, sorry about that.
good morning. We're ready for your welcome.
Yeah, okay. Give me 1 second here. So I know Willem asked to join the BGC e-call today, and sorry I was a little late. I just got caught up in another call. Give me 1 second if you don't mind, just while I set up this computer here. Really,
No problem.
my apologies. Okay, so thank you to all the participants for joining the call today. Sort of the BGC call that we've hosted on a quarterly basis, and we've made significant progress. We've completed, of course, the preclinical trials, at least the genotyping piece. From what we call the preclinical 4 results. And today's presentation is going to be from several of our participants. The first one will be around the performance of the probe variant pairs to diagnostic performance. That would be through Jeremy. The next would be Dr. Westhoff talking, taking Q&A around this. We have Bill Lane talking about blood typer and ethnically diverse BGC samples. As many of you know, that BGC samples that we processed had, we made sure that there was significant diversity within those samples. And then lastly, we would have how to translate genotypes at the 10,000 variant positions in HLA class 1 and 2, and that's through Professor Alexander Dittli at the University of Düsseldorf in Germany. So with that, I'd like to thank everybody for your attendance, and also welcome the speakers. Maybe we pass this along to Jeremy. That's good.
Okay, thank you, Shantanu. Connie,
Yeah.
Let me see whether I can figure out how to share a screen. Can someone confirm you're seeing a PowerPoint presentation?
We are, and just so folks know who Jeremy is with Thermo Fisher, and really he's the brains behind the probe sets, et cetera, and getting these this Axiom universal array going and typing. And Jeremy, I don't know how long you've been with Thermo Fisher, but you certainly have impressed us all with your knowledge of this platform, so welcome.
Thank you. Yes, I've been with Thermo Fisher in algorithms and array design for 14 years now. And I'm very happy to have been working on this project for 6 or 7 years at this point. So what you're going to see from all three of us is the culmination of a lot of work by a lot of people. I'm going to talk about my little niche in the analysis, which is really at the very low level, looking at signal. Bill will be speaking to translation, and Alex to an even higher level of integration of the data. So I think we have a nice progression here.
Hey, Jeremy, we don't see the presenter view. We see notes view. Just want to tell you the display.
Let me fix that. Thank you for letting me know. Okay, going on. So I'll be talking about tuning the performance of the probe sets on the array. This talk has 3, I hope, very short parts, a simple guide to the calling algorithm so that the next part makes sense. A few methods we're using for improving the performance of basic genotype calling on the array. And then some very recent refinements we've made to copy number calling in the RH blood group system. So first to the algorithm. At a very low level, what we get off the array is light. We get signal in 2 colors from each sample, from each of tens of thousands of probes on the array. So when we look at one variant, we typically look at one probe set, which is usually one, sometimes a pair, sometimes even more unique oligo probe sequences synthesized on the array. And what we get is for each sample, shown as a little black dot, a signal in A and a signal in B for each probe on the array, where A and B are the 2 colors. It's a little bit abstract, and I won't get into the hours and hours of discussion we could have about A and B versus wavelength. I'll spare you all that. So starting with these basic data, we can see we have in this cartoon drawing 3 very clear clusters. A group of samples with high signal in A and low in B, a group of samples with signal that's high in B and low in A, and this will probably work better with a better pointer, and a group of samples that has intermediate signal in both channels. First thing we do is transform this into a more tractable set of axes. Now, these are exactly the same data, the same 3 groups of samples, but now we're looking at them in contrast, basically the ratio of A signal to B signal versus the total signal, the sum of the 2. This just helps with the next calculations, makes it a little more tractable, also sometimes a little more obvious to the eye. So an important part of the algorithm is our prior expectation of where the data are going to fall. So I've drawn in these dashed ovals our previous knowledge, or in some cases, our previous guess, as to where we expect the data to fall. We inform the algorithm about this, so it starts looking in roughly the right place. It can range from a very gentle nudge to think that the heterozygotes are going to be around the middle of the space, to a very strong assertion that we've seen a lot of data, and that's exactly where the heterozygotes are going to fall. In this cartoon, I'm showing that the data don't exactly match our prior expectation, so we can hope we weren't too confident about it, but we're close. The next step is simply to combine the data along with the priors, our prior expectation, and generate a posterior expectation drawn in solid lines of where the data are in this data set, from this plate or from this batch of samples. So you can see these solid ovals are considerably closer to the data, matching up quite well. And then the final step in calling is simply to calculate probabilities. We say we have determined that the BBs, homozygotes, are here, described by our posterior expectation. For every single sample in this plot, we calculate the probability that it arises from this Gaussian distribution. So that's extremely likely for the samples that are right here, highly unlikely for the samples away from it. And what you see is, for the most part, we're able to confidently say this sample can really only arise from the heterozygote cluster. It must be a heterozygote. I've drawn in one little outlier here. It's closest to the red cluster, to the AAs. It's most likely an AA, according to our simple calculation, but it's far enough out that the probability that it's not is high enough that we're unwilling to call it, and we assign it a no call instead. So the important concepts here for the next parts of the talk are really that we have a prior expectation, and it guides the algorithm into finding the data and calling genotypes. So what do we do with those concepts? Our task here really is to find the best probe set for each variant and then make sure it calls accurately. So here's an example of the CX variant where the important SNP is multi-allelic. The reference space is a G, and it can become either a T, C, or A. And the genetics of this are that the T and C are extremely rare. And I've drawn these big circles of slashes over them, obscuring your view of the clusters, but essentially there's one big group of samples here with no variation. Looking at the G to C variant, there's one big group of samples, all Gs, no variation. When we look at the possibility of an A, we see some heterozygote samples. There is some variation here. And we happen to know that we're concerned with that G to A variant. Now, the algorithm doesn't know that. And part of what we do is automatically pick the best probe set to recommend each locus. And occasionally it was picking one of these uninformative variants. So all we have to do here is tell it, you're not allowed to pick those. We only want to look at G to A. And we're doing a very good job of calling them. Things get a little more complicated from here. We have the ability in the math to reuse a posterior as a prior. So once the algorithm has found a good solution and is calling accurately, our prior expectation is now updated. We've seen the data, we've seen that it's accurate. Now we know where the data fall, and we can inform the algorithm of that. To do so, though, we need to judge those posteriors and ask, is that right? Is it doing a good job? So here I have one probe set shown in 3 different groups of samples. In this first example, all the samples are being called reference homozygote. There's no variation. So there's not very much information in here, really. This is telling us, here are the homozygotes. It doesn't tell us where the data are going to fall if we see variation. In the next 2, we have samples that actually have variation. They're much more informative. But in one case, the algorithm managed to miss this group of heterozygotes. They're all no-called. You might be able to see that they're little gray boxes. So this posterior is wrong. The algorithm missed this, did not adjust to the data, and we don't want to carry that forward. But in this third data set in the center, the algorithm did what it's supposed to. It found the heterozygotes, called them very cleanly. So this is the most informative posterior and the best one. It's both based on real data, actual heterozygote samples, and it's correct, it found them. So we take this posterior, modify it in various arcane ways, and it is now our new prior expectation. And it'll inform future genotyping to make it more accurate. Next trick. When the algorithm doesn't get it, we can go in there by hand and push things around. So here's a probe set in which you can see by eye, there's a large group of homozygotes. There's a smaller group of heterozygote samples. It's clearly distinguished from the homozygotes, but because of the spacing and the very large variance on the homozygotes, the algorithm managed to miss it. And it found this one minor homozygote sample and decided, well, that must be the heterozygote cluster. It's the next cluster it found. So this is not right. We can go in by hand and just relabel things. We tell it actually the correct heterozygote posterior is right here on top of these heterozygote samples, and we'll move the homozygote posterior and then prior to be on top of the clear minor homozygote sample. Then we try again to make sure it worked, and you can see now the algorithm is catching everything. You see the dashed lines showing what we changed the priors to, and the solid oval saying, yeah, the algorithm agrees, it's finding the data and putting its posterior expectation right on top of it. So this way we improve the accuracy of calling on this probe set. And finally, this is another example of roughly the same thing, except the algorithm is doing something a little interesting here. By eye, we have a clearly distinct cluster of heterozygote samples. The algorithm is calling some of them as homozygote. They're being grabbed by this big, heavy homozygote cluster right here, and it's therefore miscalling them. And it's also missing these minor homozygote samples. What's going on here is we have a large number of parameters influencing the algorithm. One of them just keeps the 2 clusters from coming too close together. If you look at the axis here, this is very close spacing compared to the other probe sets. And the algorithm is simply prevented from finding the right solution. So The fix here is to give it a little push the same way we did on the last one, informing it, no, the data are where they are. And you can see actually we sort of had that previously. And we changed that one parameter to say, and you're allowed to put these closer together. And now the algorithm can call it correctly, and we'll do so going forward. So moving on to copy number. We have some very recent refinements we've made to calling in the Rh blood group system. The big C versus little c antigens are determined by a single base variation in the exon 2 of RHCE. The origin of this variant is a gene conversion event. The RHCE exon 2 and part of intron 2 were replaced with the matching parts of RHD. So this big C, little c variant is actually measured now 3 different ways on the array. We look directly at that SNP and genotype it. We look at the copy number of the gene conversion event. This can be indicated by a bit of unique sequence that's at the end of the gene conversion sequence. So we have this 109 base pair insertion effectively in intron 2 of RHCE. And there's enough unique sequence there, we can actually monitor the copy number of that. And very recently, we altered the configuration for the array, not the probes, just how we're using them, to directly call the copy number of exon 2 of RHCE and of RHD simultaneously. There's enough unique sequence, unique to each of them, mostly in the intron, that we can actually define this small intragenic region and uniquely call the copy number for each gene. So the task here was to determine the probes that were unique and that actually behaved as though they were only responding to one gene, and assemble those into a set that the software uses to call copy number. So this is working quite well. What I'm showing you here is the copy number of RHCE exon 2, and actually that little piece of the intron 2 as well. The graph shows all the samples in a batch, and it's showing the median ratio, log ratio of the signals coming from probes in that channel, in that region. So what you see is 3 distributions. In green, we have copy number 2 of RHCE. So this would be a homozygous for little c. 1 copy of RHCE exon 2 and 0 copies of RHCE exon 2, so this would be homozygous for big C. And it is exactly complementary to the copy number of the gene conversion. So when there are no copies of the CE exon, it's because there are 2 additional copies of the D exon. When it's heterozygous, we see heterozygous in both. And when there are 2 copies of the original RHCE exon 2, that means there are 0 copies of the gene conversion. So this is all working out quite nicely and can serve to confirm the copy number call on the gene conversion and the SNP call. One consequence of this is that we're measuring RHD exon 2. It actually has 5 different copy number states, which we're showing here, from 0 copies to 4 copies of the D exon. At 0 copies, this means we've had a double deletion of RHD, and we have 2 copies of little c, so 0 gene conversion events. At 4 copies, we have both copies of RHD still present, and we have 2 instances of the gene conversion were homozygous for big C. I'm showing you the prettiest plot. We have the resolution is not always quite so clean between 3 and 4 copies, but still we'll be able to use this information for additional confirmation and maybe discovery in the RH system. And with that, I'll pass this along to Connie for question and answer. But first, I'd like to thank the BGC analysis team in particular, Nick Liedall, Bill Lane, Ram Varma, and myself. The entire blood genomics consortium has been extremely active and busy producing all this great data.
And I'd like to give a special shout out to the Thermo Fisher field application scientists and bioinformatics scientists who were a huge part of making all of this happen. Thank you.
Thank you, Jeremy. And I think one of the strengths of this whole program within the BGC has been the informatics and the software, in addition to the diverse set of samples that we've collected. So thank you for giving that excellent introduction to the bioinformatics that happens behind the scenes. With that, I'll hand it over to Connie for Q&A. We can take questions, I guess, in the chat forum, or feel free to ask.
Yeah, thanks, Jeremy. I hope that gives everyone an idea what how your samples are contributing to the background work that needs to be done to select the best probes for calling. So this isn't, just a straightforward comparison of the data you've submitted to us with coming off the array. That information is really being used to decide which probes are going to be ones that are what we call locked down to determine the genotypes. So thanks, Jeremy. You've been instrumental to this. Any questions? I don't see any in the chat. Any questions from those doing the discordant analysis? If not, you might think some later, put them in the chat. We have kind of a limited time, so I think we'll move on to Bill Lane.
All right.
Most of you know Bill. Bill's director of tissue typing lab and of the clinical lab informatics at the Brigham. His whole research program is involved in actually doing the analysis of genomic information for blood typing. So, Bill?
Yeah, let me share my slides. All right. You see it?
Yes.
Great. All right. So thanks, guys, for letting me talk. We've talked about some of the results prior. We're going to show you some new results today. So first, a couple disclosures. You can read them there. Consult for a couple companies and the work we do here on the array. So the outline of the talk is we're going to give an introduction. Then I'm going to talk to you about a couple of changes that are sort of unique or constrained somewhat ethnically, and with a major focus on African ancestry. The first is the D3A CEVS 4 through 7D hybrid, the U negative state, Duffy weak and nulls, and then hemoglobin S and C. And we'll talk about, for each of those, a little bit primer on the genetics of them, and then about the array and its performance. So before we dive in, just so everyone's on the same page, what are blood groups? Blood groups are essentially proteins or carbohydrates that are on the surface of red cells that can differ between individuals, and that during exposure, during pregnancy or transfusion, an individual can make antibodies against these differences, and that can have subsequent implications for future exposure. In terms of the complexity within this space, this is important because over 100 million units of blood are collected each year worldwide. And if you look at the number of things we got to think about as antigens, or these immunologic differences between individuals, there are 340 of them formally recognized, or greater than that formally recognized. They're sort of grouped into categories, 43 major categories at the moment, controlled by 52 genes and greater than 2,000 genetic changes, defining the presence or absence of these different antigens on the surface of a person's red cells. So it's a fairly complex space, and also very varied in terms of the number of genes and the molecular mechanisms behind them. So our goal was to try to apply genetic testing to determine these antigens. And the way in which we're doing that is we're using, as Jeremy showed you earlier, some of this array data on these Axiom arrays, and we formed the Blood Transfusion Genomics Consortium to help funnel all this data and to work on it. And then two technologies that I've created in collaboration with Connie called bloodantigens.com, which is a database repository of curated blood grouping antigens and their genetics. That's sort of meant to be a database for research use. And then we then put onto that interpretive software called Blood Typer. And over the last decade or so, we've been moving these algorithms and these databases forward towards clinical accuracy and utility. And the array is sort of the fruition of much of that work. And so the idea is that this assay plus the interpretive pipeline can essentially give you with a readout of almost every genetically defined blood group antigen that one could determine from the genes. So how have we done so far? Well, we've published before. Nick Liedl had a fantastic first author paper in Blood Advances, where we looked at a set of over 7,000 English and Dutch blood donors and compared them to their known typing in the donor systems. And we found excellent concordance, 99.9% accuracy, for example, over these antigens for which we had a large number of results to compare against. And so the important caveat here, obviously, is that was mostly European ancestry individuals. And so today I'm going to show you a little bit of our work to try to diversify that set of ethnic samples. So first caveat, this is preliminary results. We haven't fully finished this analysis. There are still some samples to work through, still some areas that we need to get to. All of the results I'm going to show you today were run on the UBDT version 1 array, the Universal Blood Donor Typing Array. This is what we call the small array, which ironically actually means it contained more samples. We just look at less genetic changes than if we're going to do a large population GWAS type array approach, like on the other array that we have. So you can run 384 samples per array plate with this particular format. So we can run many samples, and I'm going to show you today the results of really hundreds and thousands of samples that have been aggregated down to the more important ones for today's talk. In terms of the runs, I'm going to show you some samples that are the gold samples. These are samples that came from sanguine from Africa and African American samples from the New York Blood Center. And then the preclinical 3 study, as we call it internally, or the challenge plates colloquially. These are samples that were selected by the different centers in order to cover essentially rare or interesting genetics or antigen phenotypes that we wanted to make sure that the array could interpret. And these samples were run at the New York Blood Center and at Sanguine. We have just started the analysis of the Sanguine data, but last week I was able to get a little bit of a look at the New York Blood Center data. And so I'll show you a little bit of those results as well today. So let's dive into our first example, D3A CEVS-D hybrid. It's a mouthful of a name, but it's it'll become apparent why that name is what it is in a second, hopefully. Okay, so here is the 2 genes, RHD and RHCE. These are the 2 genes that define the RH system. The D gene, the RHD gene, is importantly where the D antigen is expressed from. And this particular state is what we call a hybrid. It's where bits of one gene convert and move into the other gene. And so this particular hybrid, based on the name, you might imagine, it is when 4, 5, 6, and 7, those exons and surrounding intronic regions move into RHD. And so this replaces quite a large amount of RHD with RHCE, and has 2 important consequences. One, it actually essentially removes the serologic entity of the D antigen by doing so. Even though that protein is expressed, it removes so much of the original D gene that the D antigen no longer is present. And then importantly, it introduces the C antigen on that particular gene. And so you have a C antigen that is also expressed independently in the context of this hybrid on RHD. And that's important when we go to type for C. We need to also be able to type for its expression from this particular entity, not just the C typing that Jeremy showed you earlier. So if we look at our samples, we had 72 of these gold samples with previous typing for this particular genetic change. And so I'm going to take you through them, some of the scenarios and what we had. So we had 2 homozygotes. This is 2 samples where both the mother and the father gave a copy of this hybrid to their child. You have copies where you have only one copy of it, but it is expressed in that and fully functional because the other copy of D is deleted. And we call this the hemizygous state, meaning that one of the genes has essentially been deleted and is no longer present. So these, for functional purposes, look as if they're homozygotes on the surface of the red cell. And then in terms of the third type, we had heterozygotes. These are people who had one wild type copy of RHD and then one copy of the hybrid. And so when we looked at the array in this initial preliminary data set, we were 100% accurate at getting the first two scenarios and then 95% accurate at getting that last scenario. There was one sample that wasn't identified. So it's fairly decent accuracy, I think. This is a very challenging, complex structural event, and this was our preliminary data. And so we then had 14 samples on the challenge plate, which is the newest of the data. And here we had 2 homozygotes, 2 hemizygotes, and 9 heterozygotes, and we had 100% accuracy across all of those. So we're pretty happy with these results. We think that we can identify this change with a fairly high degree of accuracy, and we have more data coming to continue to look to see how this accuracy holds up in more samples. So let's switch to the next genetic change, and it's the U antigen. The U antigen is very important in African ancestry, in that it has some role perhaps in malarial evasion and protection. And so the way in which people look for this U antigen, its state, is by looking essentially to see if the GPYB gene is present or absent. The way most SNP genotyping assays do that is they utilize 2 SNPs that they're looking at for other regions like UVAR and the big S little s change, and those are SNPs in exons 4 and 5 of the GYP-B gene. And if those reactions don't work, they essentially say that it's U negative because they're saying essentially they failed because the gene wasn't there, and they use those as a proxy for it being present. What's interesting here is that myself, Connie, and a bunch of others, have within the last few years really identified that the genetic change for this particular difference wasn't perfectly mapped into the African background and in the ancestry. We had thought it was a deletion, this one here on the top, that only partially deletes GPYB and a surrounding nearby gene. But it turns out that it is probably a very rare Caucasian change in a particular family in France. Whereas the majority of the changes that actually lead to this difference functionally in most African individuals are these 2 deletions I show you here, below that del B left and del B right, which delete the entirety of the GPYB gene. And so we built probes across this region, just like Jeremy showed you for the big C, and we determined the copy number essentially of how many GPYBs are present. And so we're able to then detect this change. So we're using a copy number approach essentially, not just an assay failure approach. And so we think this will be a more robust approach, as well as we're not relying just on three markers to call the presence of this particular change, but rather many markers across that gene. It also affords us the ability of determining the state of one copy being deleted or both copies being deleted, something you can't do with just the SNP genotyping approach. So how did we do? Well, in the gold samples, there were 245 samples with previous U genotyping. 5 were U negative, meaning complete absence of the gene, and we were 100% accurate in detecting that. And 239 had U positive, and we were, again, accurate at predicting that. Since the previous data was SNP genotyping, we couldn't know whether that in those U positives there was 1 copy or 2 copies of the GPYB gene, but that's something that we could go back and look at with other assays we have since developed, but those would have to be run one at a time. It requires a separate PCR for each of those samples. And then we had 3 U negatives in the challenge plate that were U negative, and those were all called accurately. So we're very happy again with these results. The third change we're going to look at is Duffy weak and Duffy nulls. And so this is also another malarial resistance change in those of African ancestry. This is an extremely common, some of these changes in Africa, almost near complete penetrance in certain regions. And so very important ability to detect this accurately. And so the main change in Duffy is in the gene ACKR1, and it's a SNP position at position 125. You have either a G or an A, and that defines whether or not you're Duffy A or Duffy B. If you inherit 2 copies of the genes, one with one and one with the other, you're both Duffy A and B positive. But there are importantly some overrides to this. One is that at position 265, you can have a T there that gives rise to a weak Duffy phenotype. And this is important because that sometimes causes issues for serology, and serology sometimes cannot pick up these Duffy weak phenotypes and calls them Duffy negative by error. And then for us, in terms of genotyping, the really important position is this -67 T to a C, and this is in the UTR region, but it's a GATA promoter in that region, and there's that second change is a null state, meaning the entire protein isn't expressed particularly in a red cell context. And so this is a very highly penetrant change in Africa, and so we need to be able to get this change correct if we're going to call Duffy in those individuals. So how did we do? There were 243 samples in the gold data set with this typing, and so we had 46 that were A minus, B positive, 100% accuracy on those, and then we had A positive, B minus, 100% accurate on that. We had 16 samples that were A pos, B pos, 100% accurate on that. And then we had some samples with Duffy weaks on the B antigen, and we were 100% correct on being able to find that particular state compared to the previous genotyping. And then there were 146 samples that were A minus, B minus, and we were 99.3% accurate at detecting that. There was one sample that was called A positive, B positive by the array. So again, highly accurate results on this change. And then the final change final genetics system we're gonna look at is the sickle cell hemoglobin change and hemoglobin C disorders. And so if here's a molecule of hemoglobin here on the left-hand side, you can see the 2 beta chains here in light blue, and I've highlighted the position in each of them that define the amino acid for hemoglobin S. And so at this position 6 in the beta, in the HBB gene, you go from a A to a T in the codon, meaning a glue to a val, and that single change is enough to cause this disease. And what's interesting here is hemoglobin C is the same exact amino acid position. It's just a different spot in the codon. You change that first A and you create a lysine. And so this is important because it does create somewhat of a challenge for Jeremy when he's designing his probes, because he needs to create probes that can detect these 2 changes that essentially would be overlapping with each other in terms of probe binding. But luckily, Jeremy, as you saw earlier, really knows how to make these probes well. And there was a probe that does perform very well, and we've selected it for use here. And I'll show you those results. And so in the gold samples, there was 1,925 samples with previous HBS testing based on PCR, and 1,842 of them were wild type for the AA change. So this is the normal copy of hemoglobin, and we found that with 100% accuracy. 78 were hemoglobin S heterozygotes with then another copy of wild type, and we were 100% accurate at detecting that state. And then 2 individuals were hemoglobin SS, so sickle disease, and so we were 100% accurate at detecting that. And then what was interesting is on the array, we also found 3 samples that were hemoglobin C homozygous, and that is a change that wasn't even detected on the previous genotyping assay that had been run on these samples. So we went back and confirmed it with a different assay, and we in fact did confirm that the array correctly called hemoglobin C in those 3 cases. And then there were 33 samples that were heterozygotes for hemoglobin C. We haven't confirmed those yet, but based on the other results there, I'm fairly confident that those will bear out as well. And so with that, just lots of people to thank here, both in the development of blood type over the last decade, and then tuning it for the array over the last 5 years or so, and especially Jeremy and Nick and everyone at the consortium for their recent help in trying to get all this stuff across the line. So with that, I will end.
Thanks, Bill. There's one question I noticed coming in from Tom. Bill, I don't know if you can take a look at
Yes,
that instead of me
sure.
just reading it out.
Given the functional role of blood group antigens, is there any work or data analysis to determine whether association of blood group antigens expression, lack of expression could be linked to severity of hemoglobinopathy symptoms, frequency, and crisis event in individuals with hemoglobin disorders, which have the same hemoglobinopathy genotype? Yeah, I mean, there's definitely a lot of people associating different blood group antigens to different disease states, not just ones constrained to hematology, but I don't know of someone looking at that specific question. But that's certainly the types of things that a large worldwide data set like the one we're generating could allow for, right?
Any other questions from the group? Raise your hand or actually just speak up.
They're a very shy group today, Connie.
Yes, thanks for all that good background work on with a focus on African American samples. Would you like to just comment, please, on some of the challenges that we're going to have with the Asian samples on the glycophorin locus?
Yeah. So to me, there's there's a couple of things I'd care about in the Asian samples. One is the JK nulls, being able to call that accurately. That's a very common change in Asia. And then the other is, like you said, the glycophorins. And I think we're going to have some challenge with MIA, but I think we're going to have to look at some samples. I think there's a couple on the challenge plate, and so we're going to have to look and see what we can do to get them. It's a small structural conversion, and so we can't use the same copy number approaches that we do for these other larger structural changes to be able to detect them. So we'll have to think about another way. So. I think, Tom, you have a question?
Yeah, hi. Can you hear me?
Yeah.
Yeah, okay. Thanks, Bill. That was a really interesting presentation. With regard to the numbers of patients or donors that you type that are U negative, obviously it's quite hard to find people of that rarity in order to perfect the primers. Like, as the array goes forward, how are you building data sets to constantly improve, constantly redesign your probes, and constantly make incremental development throughout, as you see more and more people, obviously with the rarities that you're searching for? Yes, by definition they will be infrequent, but how are you making those incremental developments?
Yeah, a couple of things. Well, as we see discordances, we work them up very extensively with other technologies, and we have found a bunch of novel alleles in the previous runs of stuff, and so we will find those eventually as we go on. Specifically with regard to the U, Connie and I did a really extensive analysis of the 1,000 genomes data and another and the Simon's diversity genome data set. And we looked for other deletions or structural events in those regions, and others have done similar work. And so I know there are some rarer changes there. And I think, though, the copy number approach is the way to get at it, since it will be less prone to an underlying SNP causing a probe failure, because we're looking at many probes across that region. So if one probe doesn't behave as you might expect, the other probes, their copy number should still tell you that the gene is missing, because all the changes we saw for the most part were most of the gene or all of the gene missing, and then just different intragenic regions. So I think that the approach we're taking will be fairly robust with regards to other deletions of the GPYB gene. That said, this is a very complex genetic space, and I don't think that we've fully have appreciated how many things can happen in that region. But there are like some weird events where some of those genes are duplicated and then inverted. So things that we don't see, we haven't really characterized in any of the other blood group systems, and nor really have ever had formal characterization of those in the blood group field. These are just genetic studies have found these very unusual individuals.
That's great. Thank you very much.
Could I ask something maybe on the same lines that Tom was referring to? So first, you and Jeremy gave very nice, clear talks. It was really good to understand. As I know the probe sets that's going to go for licensures probably can't be revised later, but say once you find some of those rare, say homozygotes or even heterozygotes, do you have the ability to modify then the calling and change, the algorithms?
So from a technical standpoint, absolutely. That's a lot of what Jeremy and I and Nick and everyone we're all doing right now is it's the homozygote rare states that you likely are going to need more work on because you probably will have exemplars of the others at this point in this amount of data. I don't know, Connie, can we change the posterior, like those things that Jeremy was tweaking earlier after you get the approval, or?
Well, I think this is one reason why Thermo Fisher and wants a RUO platform, certainly to start out with, because you have a lot more flexibility of adding, subtracting, etc. So it's a good question. We hope going forward that certainly in the US, FDA licensure will realize that, this can't be a hardwired area, but a lot to be determined yet in that as far as regulatory approval, which is different in every country, certainly.
Yeah, you certainly
Matt,
wouldn't want to lock down an assay and have a worse assay for it, right?
Exactly. So how much flexibility you're going to have modifying is still to be determined. Matt has his hand up. Go ahead.
Yeah, hi. So Matt Hazel, NHSB TRCI. Just a quick question. Do you think there's opportunity in relation to these probes in the future for them to be translational? So treatment of sickle patients and things, transformation of the HBS to HBA, that sort of thing moving forward, maybe using CRISPR and things. Is it translational for that? Are these probes that good? Could they be used for treatment?
You mean like changing someone's genetics, or?
Yeah, so CRISPR is sort of it's on the horizon, isn't it? Of future treatment of patients, things here. And you've got what appears to be some fantastic probes, because to be able to pick up the differences in those hemoglobins in the sickle population and things, and the ordinary adult hemoglobin populations and things for that in your testing. The ability to anneal and things to people's DNA and things like that. Is there a potential here for this to be somehow translational in the future, maybe for treatment of patients of this type?
I'm not sure. I'll say one thing that Jeremy I don't think mentioned on is that these probes are kind of interesting in that there's one probe that binds and then a second probe that binds, and there's a ligation step in between them, and so it's not just one probe. There's a lot of dynamics there in order to get that specificity.
Yeah, because I can imagine in that sort of treatment, there's obviously so many cells in the human body, dose-wise and things and stuff like that. It might not be a one-hit treatment, if you know what I mean. You might have to have multiple hit treatments, in which case you're not going to want that probe annealing to an area you've already changed, if you know what I mean. So you're going to want to have specificity in that heterozygote sort of patient state, maybe, and then maybe there's a tipping point where you get them to the point where you tip them over, so maybe they stay heterozygote slightly, but they're more HBA after treatment than HBS. Do you know what I mean? It's sort of trying to visualize the future. So just wondering, that's all. So just sort of put that out there.
I'll leave that to Shantanu to figure out whether he can create a new market from that. He's got excited based on your question.
I would say, you probably want to rely more on the next-gen sequencing to get that level of specificity and accuracy rather than rely on this probe. Obviously, there's quite a bit of more discovery work that could be happening in later phases. So the array is a snapshot of what we know today, obviously.
Yeah, I just felt like it aligned very nicely with that future state that I think we're heading towards, realistically, with other disease states and things and stuff like that. So, or disorder states, I should say. So thank you very much for answering my question.
Yeah, there's a question in chat about whether or not serology, it says, anti-sera for serology is independently run to confirm the antigen. Do you anticipate the array should be run twice for reconfirmation? Again, Shantanu would love that if you ran the array twice. So Connie, I think you've thought a lot about this. What are your thoughts on that?
Yeah, so in the US, certainly if it's a licensed assay, it's a test of record after 2 tests. So are we talking about replacing serology here? Yes, we're actually very comfortable in the US doing that with the assays we're doing now. and it's working very well because they've been highly validated with serology. And so that's the idea here, to have an assay that you can use for clinical use. One thing I do want to say that was brought up kind of with Matt's question, we were being very careful to indicate that this is an array for blood typing, not for disease diagnosis or disease-related, care, etc. So it's for blood typing to clinical scale rather than any disease-related associations. So,
yeah,
but very good questions.
Hi, I have a question regarding the missed calls. Do you have an idea why do you see this? Is it stochastic or systematic? And the question is, let's say for this heterozygotes with the hybrid RHDCE, if you rerun that sample, will you get the same signal, or will it be closer to the cloud where finally you'll be able to call it correctly?
Yeah, we're still at the phase where we're kind of just collecting the data and then go back. We generally hope that things like that, that it's something where you can fix it in the software. Like as Jeremy showed you, the clustering and the selection and things like that. In terms of whether you get the same result each time, yeah, if we don't change any of the stuff that Jeremy's talking about, you should theoretically get the same result, or at least we seem to get the same result every time with that data. But we can have high influence on its especially the early pre-processing steps of whether or not something's present or absent. We have typically in the past done, this is the work that right now is being done at Sanguine and New York Blood Center. They're going and pulling all the samples and repeating serology and genotyping with other assays and things like that, so that we can get a sense of whether or not there's some other, as mentioned earlier, maybe some really complex genetic event underlying that, right? That caused an issue. Or was it just the probe was in a boundary region, for example, like Jeremy showed you, sometimes things are in between regions. And so you could imagine if the null probe was in between regions, right, that it would have been not called, and maybe that's why that sample didn't get the null, right? So the first thing to do is to confirm with genotyping that the null is actually present and which null is present, and then to look to see why it wasn't found on the array on the data.
Thank you.
Yeah.
And I will say one thing more to you, also to your point about if you ran that sample again, would you get the same result? The same samples are being run in the Netherlands and in New York. And then the same set of probes are on the UK larger array. So these probes are actually being tested on the same samples in 3 different sites. And then we're doing a comparability, and that data is still being analyzed, but peripherally it looks excellent. That there's great reproducibility doing this assay at a different site, different time, different conditions. And that's an important for the assay also, reproducibility and repeatability.
Yeah.
Thank you for that question.
Yeah. Guess if there are no more questions, we can move to the next presentation by Professor Alexander on how to translate genotypes at 10,000 variant positions into HLA class 1 and class 2 antigen types. Thank you, Dr. Alexander.
Hello, and thank you very much for the nice introduction. I will now share my screen. Could you guys kindly confirm that you see my screen?
Yes.
Marvelous. Well, hello, and first of all, thank you very much to the invitation, in particular to Willem, to Nick, and of course to the whole BGC. In the following, I'll give you a brief introduction to how statistical HLA type imputation actually works. I will try to keep this not too technical. However, if you have technical questions, obviously I'm very happy to answer them afterwards. And the title of this talk is how to translate genotypes, and that is SNP genotypes at 10,000 variant positions into HLA class 1 and class 2 antigen types. So the first concept that I need to introduce and that most of you will be familiar with is the concept of linkage disequilibrium. So if you want to remember two things about the major histocompatibility complex, A, obviously it is very diverse, and secondly, it is characterized by strong patterns of linkage disequilibrium. Actually, it is one of the regions of the human genome that exhibits the strongest patterns of linkage disequilibrium. That has actually been appreciated for quite some time. What you can see here on the left-hand side is a plot from a paper from 2004. What you basically see here is a matrix that shows you SNP positions at the X and at the Y axis. And the color of the different dots here tells you something about the correlation structure of the SNPs at the corresponding positions. So speaking from a statistical perspective, what this basically means that whenever you have a red field in this plot here on the left-hand side, that the genotypes of the corresponding variant positions are correlated with each other. You will notice the strong correlation structure across the diagonal, and that is quite typical. So nearby SNP positions are obviously correlated, but you also see points and dots off the diagonal, which shows you the so-called long-range haplotype structure of the major histocompatibility complex. In effect, or in consequence, what this really means that if you know the genotype of a few positions along the MHC, you can make a very informed guess about the genotypes of other positions along the MHC. That's the statistical interpretation. The biological interpretation is that we observe low recombination rates across that region, that basically biological recombination does not tend to break up the chromosomes in these regions very often. Now, can we use this for statistically inferring the HLA types from SNP genotypes? And it turns out we can. And a nice way to see that graphically is as follows. So what I'm showing you here is on the left-hand side, I'm showing you SNP genotype plots. That basically means that each line in these plots here is one individual chromosome. And the different boxes tell you whether at a given allele we see the majority or the minority genotype. So basically, this here is one haplotype. And on the right-hand side, what I'm showing you is the HLA genotype here for HLA-A for that particular haplotype. You can see that if you compare the SNP haplotypes of the different alleles here, these exhibit different and clustered structures. So if you now have an additional chromosome, perhaps without HLA typing available, you can now compare this new haplotype to the existing haplotypes and ask which of the existing haplotypes is this new haplotype most similar to. So in summary, it is well appreciated that different HLA alleles exhibit distinct SNP background haplotypes. But as you can see here, even within a single population, these backgrounds are not strictly homogeneous. So you can see patterns, for example, these block-like structures here. You can see that these block-like structures very clearly differentiate between the alleles, but you also see that there are differences within the groups. And so alleles have heterogeneous backgrounds, and once we move, for example, to cross-ethnic HLA type imputation, these differences become more pronounced. So let's try to state the HLA type imputation problem a little bit more formally. So the type of data that we're dealing with is basically data from chromosome 6. And we're dealing with diploid data that is phased or unphased. And we're dealing with individuals that are genotyped at given positions by a SNP genotyping array that are also genotyped at the HLA loci of interest. Of course, we're not dealing with only one such individual here, but with multiple individuals, often thousands or tens of thousands. And we refer to these individuals collectively as the reference panel. From that reference panel, we want to build a statistical model of the HLA alleles and their surrounding SNP haplotype structure. And now, with additional individuals for which we only have the SNP genotypes, but no HLA genotypes, that is basically the inference data set. We want to use the model that we built from the reference panel to infer the HLA types of these individuals here. Now, how does this actually work? In the software that we developed called HLA-imp02, we create this model, and that model takes the form of a haplotype graph. An example haplotype graph is shown in this plot here, and of course, this is not a real haplotype graph, but this is for illustrative purposes. We infer this object from the reference panel that you saw on the previous slide on the left-hand side. And so why do we call this a graph? Well, mathematically speaking, this object here consists of nodes. These are the dots, and edges that connect the different nodes. That makes it a graph. If you look at that object very closely, you will observe a number of things. One important thing is that these edges here are actually labeled. So in this case here, the edges are labeled with nucleotides. And in this column here, the edges are labeled with HLA alleles. And each level of the graph, each basically vertical column also carries a label that either identifies the corresponding SNP position or the HLA locus that we're dealing with. And in addition to all of these labels, you also have a little probability attached to these edges here. And this probability basically mean that if you are in that node here, you have a probability of 80% of moving here and a probability of 20% of taking this path here. And basically, if you start somewhere here at the left-hand side and then move through that model according to these probabilities, you can use this model to sample novel haplotypes for the HLA region. And where does this representation of the HLA haplotype structure actually come from? Well, it comes from an earlier model called Beagle that was developed at Browning and Browning, which we adapted and improved for the MHC application. And that model was inspired by earlier machine learning models for representing language. Now, of course, we're not only interested in representing the haplotype structure of the region, we also want to make inference. And for inference, we convert this object here into another data structure called a hidden Markov model. And we can condition that hidden Markov model on the observed SNP genotypes for an individual without known HLA type information, and then use a variety of algorithmic approaches to basically calculate the probability of all the different ways of traversing that model. But then basically summing up the set of possible traversals of this model and the corresponding probabilities, here at this locus of interest, we obtain a probability distribution over possible HLA alleles for that individual. And one important property of this model is, this is also illustrated in this little plot here, that alleles can appear on multiple SNP backgrounds. For example, here the DRB1501 allele appears in 2 different paths. And so therefore, this model is very well suited to, for example, modeling the HLA haplotype structure, even in the case of an ethnically heterogeneous data set. Now, how well does this work? You guys carried out extensive validation of HLA type accuracy in the BGC cohort. So you have the numbers that you have are more up to date than my numbers here. What I will show you here is older data from the original publication of the model, basically to give you a feeling for what the factors that influence the accuracy of the model are. So for example, here, let's focus on these 2 columns here. That gives you the accuracy of HLA-imp02 compared to HLA-imp01. This was an earlier algorithm that used a different approach. We're looking at European data here. You see that the performance for the European data is typically in the high 90% range. And we're looking at 2-field resolution. DRB1 in this experiment here is a little bit of an outlier. DRB1 in general tends to be a little bit more difficult to impute than these other loci here. You can also, I just mentioned that basically the model gives you probabilities. So if you only evaluate probabilities, or if you only evaluate imputations that you have a reasonably high degree of confidence about, here a 70% posterior probability, then increase that the accuracy of the model substantially, typically at a modest decrease of the core rating. Now that was on an old reference data on European ancestry samples. If we took that old data and applied it to non-European data, we saw observed reduced accuracies. So we carried out some evaluation of Asian background samples, of African and Black African American samples and Hispanic samples. And you basically see that the accuracies here at 2-field resolution are reduced, sometimes more, sometimes less, the extent of which is determined by the match between the reference and the invitation panel. However, even using this old data set, the accuracies at 1-field resolution were still often quite decent. For example, you see this like 100% column here in the African American samples here, and also in the Hispanic samples. And now it is very important to conclude, or not to conclude, that basically imputation in non-European ancestry samples is more general and difficult, is more difficult in general. What I'm now showing you is performance evaluation data using the HLA-IMB02 algorithm, but using a reference panel that was constructed for the VACGene project that was led by Alex Manzer. There's also a very recent preprint of that. And basically, so for that project, an African imputation reference panel was constructed called the VACGene reference panel, also with slight improvements to the algorithm. What you basically see here, so this is all African data. You see that the accuracies for most loci are really at the in the upper 90% range, and this is now also at full 2-field resolution. So the takeaway message from this is really that the quality of the reference panel and the match between the reference and the inference data set are the most important determinants of imputation quality. Now, of course, there are two important follow-up questions. So how can we build improved reference panels? And are these kinds of approaches built limited? Are they limited to the classical HLA loci? Can we also use them to look outside of the classical HLA loci? And in that context, I would like to highlight a recent development. And that recent development is that using long-read sequencing, we are now able to resolve full MHC haplotypes at the complete sequence resolution. And so we just put out a preprint out where we basically, for example, complete the alternative MHC reference haplotypes of the human reference genome. I will not go into this, but for example, one exciting thing about this is that this includes the full complete sequence representation of one of the haplotypes that carry neither DRB3 nor DB4 nor DRB5 allele. That will also be out soon in the journal HLA. We can basically use these new long-read sequencing technologies to build sequence-based models of the structure of the MHC. And these sequence-based models, also class of genome graphs, can then be applied to cohort-level Illumina sequencing data. And using these improved approaches for the interpretation of genome sequencing data will give us full resolution HLA type reference panels for large populations, which we then can carry and use for array-based imputation. This is basically, I think, how the loop is closed between these long-read sequencing technologies, Illumina cohort sequencing, and then bringing the information from these cohorts into the millions and millions of samples that have been array genotyped. And with that, I'm at the end of this brief presentation. I would like, so we have multiple papers, and I would obviously like to acknowledge our co-authors, but 3 people in particular need to be highlighted. We were basically partners in crime in developing these approaches, and these people are Stephen Leslie, Gil McLean, and Peter Donnelly. If you have any questions, first of all, also thank you very much for your attention, and if you have any questions, I'm very happy, of course, to answer them.
Thank you for that excellent presentation. Questions? We've got quite a few HLA experts here on the attendance. And while we're giving people time for questions, are we using the VACGene reference panel, Thermo Fisher, for 2-digit calling from the array?
No, not currently. So that is a very new reference panel, and that has not been integrated into the Thermo calling software yet, as far as I'm aware.
Yeah, that is correct. Maybe Jeremy, you can talk about the HLA typing software. That would be great, and how, and
Yeah, certainly. Working with Professor Dilthey and his colleagues some years ago, we implemented the HLA-IMP02 algorithm with an older graph, a little more European-centric than the vaccine, much more actually. And that software is freely available, well integrated into our software for genotype calling, and is in fact part of the automatic pipeline we're building for blood type calling. So that pipeline will integrate pieces that currently exist into one smooth workflow, from genotype and copy number calling to HLA imputation to the current project, blood typer integration into the whole thing.
Thank you, Jeremy.
So we have one question in the chat. How well does the reference graph-based approach deal with novel alleles? Will it try to force the results to match known types?
So calling novel alleles from sequencing, from whole genome sequencing data is still a little bit of a challenge. We're currently developing algorithms that should perform much better at detecting novel alleles. The current software, for example, out of my group, yes, it will basically give you a probability distribution over known alleles, very much similar to HLA type imputation. However, we are basically working on, once we have like a best guess set of inferences for a given individual, we're then working on basically realigning the reads to that best guess and then detecting additional variation on top of that. And we're basically hoping to complete that development reasonably soon and then basically update our software.
Thank you. And I remind everyone that the goal of the array for donors is to do 2-digit calling, not to do high resolution HLA. Anthony, please go ahead.
I just wondered what the ethnic mix of the original reference panel was.
Was that the VO2? Jeremy, do you remember if we had, I think it was mostly European, some African and Asian populations?
Yeah, actually, I think Professor Dilthey is the best one to answer this, but I think the accuracies Professor Dilthey showed are from that panel that we're using in our current software.
Okay, I've just a supplemental question. How would it cope with someone with a mixed heritage? So if their mother was African and their father was Caucasoid, would it struggle? And how would it cope with haplotype loss?
It would depend to the extent on which basically the two source haplotypes are represented in the original panel. So that what is really difficult for any kind of imputation model are basically haplotypes that themselves represent a mix of different ethnicities. But as long as basically the two haplotypes are, well, not in themselves also, well, a mix, a recombination, a recombined haplotype, that shouldn't be a big problem. And sorry, what was the second question?
If someone had haplotype loss due to disease or some kind of treatment, that's unusual for a donor. Obviously, most of these samples you see will be healthy donors. I just wondered whether it would cope with that type of sample.
It would depend on the SNP genotype input data, but most likely I think such a sample would be imputed as homozygous across the complete region.
Okay, thanks.
Alexander, we had a discussion the other day about which IMGT data set you were using for your imputation software. And we know that this data, a new version comes out virtually every quarter year. Do you have plans to keep up with these changes? And how far behind the current IMGT data set are you at the moment?
So certainly a few years. Also, the input data that are used to reconstruct the graph are not fully homogeneous with respect to the IMGT version that was used to derive these graphs. I think there will be an update to the reference panel at some point, but probably that it's unclear on what timescale specifically that will happen, because I think before we really approach updating the graph, we want to sort all the novel allele calling from sequencing-based data. And then, as I showed on my last slide, basically take large whole genome sequenced cohorts and then use these to really update the graph. And then there would also be, I think, a pipeline for regularly updating the graph, which would then probably represent G group allele levels, or perhaps even for resolution types.
Excellent, thank you.
I have a rather naive question. Since there are many ethnic markers also on the array, are those markers at all considered when imputing HLA types, or is that an independent function? Or I'm not sure my question's very clear.
I think it is. So the HLA-IMP algorithm does not take any any orthogonal information on sample ethnicity into account. So it may be feasible, for example, to use additional markers of ethnicity, for example, to then select a different reference panel for your sample, but it turns out that the algorithm itself is quite good. This is basically the graph-based thing and the presence of multiple paths, that the algorithm itself is quite good at dealing with heterogeneous input data sources. So probably in any case, you would be better off by mixing or by throwing all of your available reference data into one large panel, than letting the algorithm do the work of figuring out basically which haplotypes match best.
Thank you. Any other questions? Don't see any other hands. Shantanu, anything else? Lindsay?
No, I just a couple of closing comments. Some fantastic, you know, everybody saw some of the fantastic work that the large groups have been doing, and our goal is to make this blood typing array available to everybody. So as we go through the verification and the analysis, we appreciate everyone's patience. The results will be made, from the preclinical will be available later this year. There's certainly quite significant amount of effort from everybody. I'd like to, from Thermo Fisher, we'd like to really thank everybody that's involved in this process. And then finally, for today's speakers, very much appreciate all the contributions as well as today's presentation. I think the questions were fantastic. Really like the interactive questions that came in from the audience. And we look forward to the next presentation and educational seminar.
And also this one will be uploaded, and I will send you all the link on our BGC YouTube site.
Yeah. Thank you, Lindsay, for organizing and coordinating the Zoom call.
That's okay. My pleasure.
Thank you, Lindsay.
Thanks, everybody.
Thank you, everyone.
Thanks.
Bye-bye.
Bye.
Bye-bye.